Hello students, now we are going to discuss the next topic, that is the gene therapy from the chapter Biotechnology and its applications. What is gene therapy? Gene therapy is a technique in which we can replace the defective gene by a functional gene. As you know, in the human beings, genetic material is mainly the DNA, and DNA is made up of the nitrogenous bases that will make the codon, and each codon is specifically code for a protein. But what would happen that when a particular gene is not working, that means there will be a deficiency of a specific protein, and that protein and lacking of that protein will adversely affect the human body. So why don't we develop a technique that can, if any such type of problems will be arise, then we can replace that defective gene by a functional gene, and that technique is known as the gene therapy. Gene therapy is also used to treat the number of hereditary diseases. Hereditary diseases are diseases that will pass from generation to generation, like the hemophilia and phenylketonuria. Like such case was performed, first clinical gene therapy case was performed in the 1990 in the 4-year-old girl, that she was suffering from the SCID, means severe combined immunodeficiency. That means there is a deficiency of immunity, and that girl is suffering from a number of diseases. How this disease will occur? SCID will occur due to the deficiency of an enzyme that is known as adenosine deaminase enzyme, and this enzyme is required for the proper functioning of the T cells. And as you know, T cells will provide us the cell mediated immunity. But what would happen if this enzyme is not produced? That means the gene that is producing this enzyme will be defective. At that time, there will be deficiency of enzyme, and as a result, there is a lack of T cells in the human body. So there will be a decrease in the cell mediated immunity. So immunity will be lower down, and if the immunity of a person will be lower down, the person will be susceptible for attack of a number of pathogens. That is why this disease is known as severe combined immunodeficiency disorder, because that person is susceptible. A number of pathogens can attack in the body. Why? Because immunity is not working against the pathogens. So what are the treatment? First treatment, bone marrow transplantation. Why this treatment is feasible in this condition? Because all the cells of the blood is formed inside the bone marrow by the process of hematopoiesis. Yes, so that means the genes that is producing this enzyme is also found in the bone marrow, and that gene is effective. But if we will transplant the bone marrow, then effectively that gene will start to make the enzyme, and that disease will be cured. Second, we can use the enzyme replacement therapy also. We can culture the lymphocytes, and then we will introduce the adenosine deaminase gene in that lymphocytes that we have cultured in the laboratory, and then introduce into the patient. But must keep in mind that this type of treatments are not for long time, because T cells also have some lifetime period. After that, they will be dead. So what in these cases, profusely we need to give the T cells that have the ADA gene time to time to the patients. But how we can permanently cure this type of disease? Yes, permanent cure is if we will insert the gene in the bone marrow of the embryonic stage. That means if we found a case that the disease occur in the generation in the specific person, and in that family new member is coming, so what we can do, we can insert the gene into the embryonic stage in the bone marrow, and actively it will start working as the embryo will grow and develop into the fetus. This is a permanent cure. So gene therapy presently, if we see it in 2020, it is not working 100%. Why? Because human sequencing is not done till now completely, but when human genome sequencing will be done, then it can be applicable everywhere. So second is second topic of the biotechnology and its application is a molecular diagnosis. Yes, biotechnology is used to diagnose a number of diseases. Like molecular diagnosis include the PCR, that is polymerase chain reaction, ELISA, and recombinant DNA technology. So these are the three practical applications of the biotechnology First, we can discuss that is the PCR. So I have mentioned here. PCR is the polymerase chain reaction by which we can make the multiple copies of the specific DNA, or that DNA may be of the pathogen. Yes, we can take out the sample of the patient, and then we can take out the pathogen, and then we can isolate the genetic material of the pathogen, then make the multiple copies of that pathogen. And we are already having the samples of the number of pathogens in the laboratory, then we can match with them to find out the specific disease. So I have mentioned here it is used to diagnose the disease. But main question arises, whenever any disease will occur, and how can we find out if we are suffering from a disease? Only when we will develop the symptoms. Yes, symptoms, and symptoms will be arise only when the concentration of pathogen will be high in our body. Yes, there is a portal entry and portal exit for the pathogens. The pathogen from where enter in the body is known as the portal entry, and from where they will move outside is known as the portal exit. So when concentration of the pathogens will be high, then we will develop the symptoms, and then we will go for the checkout. At that time, for going to the doctor, we are suffering from and we are showing these symptoms, then may please tell us the disease and can help us in the treatment also. Then doctors, what they will do? They will take out the sample, then take out the pathogen and DNA. They will isolate and then multiply with the help of PCR, and then match with the samples and take out and find out the pathogen of the disease and the name of the disease. This type of technique is nowadays is used for to find out the HIV, that means for the AIDS. And second is ELISA. ELISA is enzyme linked immunosorbent assay, and this technique is mainly work on the principle of antigen antibody interaction. Any of the pathogen that is entering into the body is known as the antigen that will cause the disease, and for every antigens a special type of immunoglobulin body is produced by our body, that is known as the antibody. That means antibodies are produced. Antibodies are specific for the specific antigens, and that are produced only when the antigens will enter. So this technique will work on the antigen antibody interaction. How it will work? There are 5 types of antibodies are produced in our body. Then what they are doing during the labs, what the lab technician are doing in the labs? They are just taking the test tubes, and in test tube they are taking the samples, and that sample have the antigens. Then they will add one, they will mark the tubes one by one, and then add the specific antibodies like in IgA, IgG, IgD, IgE, IgM in one tube. Then they will found that a specific antigen antibody complex will form only in one test tube, not in the others, because only one type of antibody will be produced against a specific antigens. And if antigen antibody complex will form, it will change the color of the sample of the test tube. Then they can easily distinguish that what type of antibody will be produced against a specific antigen. Now, third technique I have mentioned here, recombinant DNA technology. Yes, in recombinant DNA technology, we can use a probe. What is the probe here? Probe is a single-stranded radio-labeled DNA. So, maintenance CBSE will ask also define the probe. Single-stranded radio-labeled genetic material, either DNA or RNA, is known as probe. And this probe that will hybridize with the complementary DNA of the clone cells, that can be easily identified with the help of technique autoradiography. So what we will do, the clone cells that have the mutated gene will not appear on the photographic plate. So these type of probes can be artificially produced in the labs for checking out the specific disease. These are known as the more practical application of the molecular diagnosis, or these are the application of the biotechnology. Thank you.